Hello friends, in this video we will explore the scaling and identification of variable features of the Seurat pipeline. Okay, and once again we will explore why we need to do scaling and how to do it. Okay, so far what we have learned, we explored like how to load the cell ranger output to Seurat object. Then we talked about the quality check and pre-processing, and then next we explored the normalizing the cells. Okay, so in this video we will learn about scaling and why we need scaling and what is actually scaling. First we'll see then why we need it and how to do it, and also why we need to identify the variable feature genes, okay, or HVF, or highly variable features. Okay, so first of all, why we need to do scaling? It is same as the concept of normalization. So we saw that why we need to do normalization, because each cell is different from each other, because of different reactions and different technical variations we see in the cell to cell variations we see. So to remove those technical noises, we are doing normalization. Now you write like the normalization goes in this direction, right? So that means we are normalizing every cell, huh? Okay, while the scaling will go horizontally. So that means you are scaling the gene across different cell, okay, so that you can compare gene to gene. So in the normalization, you are normalizing cell such that cell to cell difference you are now ignoring, or making all the cells on the same level so that you can compare cell to cell. While in the case of scaling, we are making sure that gene to gene can be comparable. So in the same example what I gave, right, if you mix and kids with adults in a running competition, it would be a bias, right? You cannot mix them. Similarly, if a gene since each cell is different, so the gene expression count of the gene 1 and gene 2 will be non-comparable because of those variations between the cell. So we need to also make sure that each gene are brought to the same level, okay, so that we can compare them. Okay, that is one region. And second region is like in the downstream analysis, when we do principal component analysis, okay, as well when we will be doing some heat maps. So these two techniques, PCA and heat maps, here dominate. If a value is very high in number, right, so those will be dominate the lower value. Like the same example, adults will dominate the kids, right? The same. So for example, this gene 1, the count is let's say 1000, 2000, 3000, 4000, in that range. Whereas in this gene, the count is in the range, let's say 10, 20, 30, 40, in this range. See, means this gene, the expression of this gene is low across all the cell, but the range is very less. The range is important, okay, 10, 20, 30, in that range. But let's say this gene is in 100 range, like 100, 200, 300. But this gene is in 1000 range, 1000, 2000, 4000, like that. So now when we mix them together, this gene where the count is very high number, like 2000, 3000, that will dominate the lower count. So that means the effect of this low count genes will be neglected. Okay, so that's where it is very important to make sure that you bring all the genes on a same level, or you scale them, or you bring them into a single scale. Okay, so scaling we will be doing in horizontal. So we'll be applying on every gene. So very simple how it works. So basically we do general standard normalization. So like let's say you do, let's say start with gene 1. Okay, you calculate the mean expression of the gene across all the cell. Then you calculate the standard deviation. Then you subtract that genes with the mean, and divide that with respect to standard deviation. So what happen when you subtract with the mean means, if the genes expression is higher than mean, okay, so those will become positive. And if the genes expression is lower than mean, so those will become negative. So some genes will be less than mean and some genes will be more than mean, right? So you will have centering at the 0. So that way, this statement is important, that when we do x minus mean, okay, so that means this will shift the expression of each gene so that the mean after scaling will become 0. So that we can now identify which are the genes which are less than the mean and which genes are above the mean. So like that. So this is generally called as centering, x minus mean. So for in this example, let's calculate the gene 1. The count is 3 to 13. Okay, so if you calculate a mean, so 3 plus 2 plus 13 by 3, the mean becomes 6. And if you calculate the standard deviation, standard deviation becomes 6.08. So what we need to do, we need to divide 3 minus 6, that is the mean, and then divide with respect to standard deviation. So when we divide with respect to the standard deviation, what will happen? The scales the expression of each gene so that the variance across the cell is 0, or variance of the standard deviation now becomes 1. So our mean will become, so after scaling, when we again calculate the mean will become 0, and the standard deviation, okay, of that will become 1. Okay, so we will be doing the same to all the genes. So this will help so that all the genes become on a same level. So you see the statement, this step gives equal weight in the downstream analysis to each gene, so that highly expressed gene, means those having very high count, do not dominate in either PCA or let's say heat map. Okay, so it is very important that we will be doing the scaling this gene. But now just see that how many genes we have. 13,000 genes in this example, and in this demo example, number of cells were less, but in reality cells will be very high number. But now so many genes we cannot do the downstream analysis and do the scaling. It will be computational intensive. So why to do so many genes? Why to analyze all the many genes? Okay, so then the concept comes like, why not I filter those genes? So if a gene is not varying across cell, so then those genes are not important to me, right? Because my objective in single cell data is to find out, is there any subtypes are there or not, like let's say cell subtype 1, subtype 2, subtype 3. But let's say take this gene 1, this is equally expressed in all the cells. That means this gene is not contributing for subtyping. So that means if it is constant across all cells, that means the variation is 0. That means this is less variable. So I am not interested in such genes which is less variable or which is more constant. So I am interested in what? Which is highly variable, means those genes is in some cell it is highly expressed, whereas in some cell it is less expressed. So I am more interested in those highly variable genes. So I will first identify which genes are the most differentiating or most variable, and then I will be applying scaling on that, so that it will save my computational time. Okay, so that's why before scaling, what we will now do, we'll first identify the genes which are highly variable, and once we identify them, then we will scale only those highly variable genes. And this highly variable genes is also important in the PCA, because PCA also, when we do in the downstream analysis, we will not do PCA on all the genes like 13,000 genes. Instead we will be using these highly variable genes. So that's why identification of HBF, highly variable gene, is an important step in the pipeline. Okay, cool. So now we will do practical here. So last we have done up to what, we did the normalized data step. So that means we did the normalization and we saw how the normalization changes. So now first we will do the identify the this, right, variable genes. So the function is find variable features, where the first parameter you pass the object and you choose a method. What method we will be using for this identification? So this method is generally variance stabilization transform method. Okay, so let me talk about that. So I'm again going to the Seurat tutorial here. Okay, so this function, let's click here. So okay, so this is the method, VST method. Okay, so basically what it does, like it first draws, it tries to I mean, what is this? So it's a plot between the expression, the mean average expression of that gene, okay, in that cell, okay, in all the cell, that genes expression versus the variance, right? So two parameter we are estimating, right? For every gene we calculated what its mean and its standard deviation, right? So we that plot. So here each dot is not cell, here each dot is one gene, and the x-axis is the mean expression of that gene, and y-axis is the standardized variance. So now you see a plot between mean versus variance. So now in single cell data, mean and variance is dependent on each other. So we see that there is a trend in it, right? So using this, a model is fit, and that using that model, it tries to scale that observation. So the method is VST method. So where you see that it fits a line to the relationship between log of variance and log of mean. Okay, so mean and variance relationship. So mean is the center point, whereas this is the spread, like how much variation is that there in the data. So between these two, it tries to fit a line, and then it standardize. Then based on the fit equation, what we get, based on the fit, it will re-modify the values. Okay, and so now basically, why we need to do identical, means why we identify highly variable features, is because we are most, let me read, I am just what is that point, yeah, yes, that's what, right? So this statement, if you see, so their procedure is Seurat described in detail here, and it improves on previous version by directly modeling the mean variance relationship. This is important. So mean variance relationship is inherent in the single cell data, which is what we see, and based on that it fit a line, and using that line it scales the observations. Okay, so the function is, and by default we say that give the top 2000 variable genes. Okay, so we can change this parameter as well, but higher means, if you retain higher number, means more computationally intensive it will be. Okay, so now let's click run this. So it is now finding the most variable genes, and it has identified. Now if you remember, right, we had a slot, let's pbmc at the rate essays, and then within that we have RNA-seq, and we had which slot, variable dot feature. So this slot, if you come here, okay, so this slot will contain, I'm just showing you the head, the most variable genes. Okay, and if you want to see how many we got detected, so we can check the length. Okay, so that is 2000, and I'll just show you over how we can get. So in PBMC object, then SA, then RNA, this is the first slot, this is the second slot, and come here the variable dot features. So this slot was empty before this analysis, but after this analysis this got filled with the 2000 gene names. Okay, just if I click directly, you see I'm just showing. So these are all 2000 genes, these are the most variable genes we see. Okay, so we can find which of by using this navigating through the object, or there is already a function is the variable features function. So this will return all the variable. So we can store the top 10 using this variable top 10 genes. Okay, so these are the genes which are top 10 most variable genes in our data set. Okay, so now we can draw a that plot what I have showed you, right? Same plot only it will come. So let's redraw. Okay, so the function is variable feature plot. Okay, so this will draw the the x and y of this one, the plot between variance and the mean. Okay, so okay, this is a old plot actually, so my bad, my system is slow again. Okay, so let it run. Okay, meanwhile I will show you. Oh, okay, so I have stored it, my bad. Okay, so we need to print it. Okay, so when we say plot 1, so now it will draw. Okay, oh, I don't know why it is so slow. Okay, anyways, I will explain, let it run. So in this you will see that I am drawing this. So in the same plot, but here I am highlighting the top 10 genes. Okay, so strange. It is not working. Okay, I have to restart maybe. Okay, anyways, so the command is simple, like you can rerun. Okay, so you will get the same plot only. Okay, but next what we do, so now we have identified which genes, not top 2000 genes we have identified, okay, which are most variable. So in the next step what we will do the scaling. So for that simply we will be using the ScaleData function. So this will only scale those 2000 genes. Okay, so now we will see, now I have drawn just to show you the box plot kind of things. See, now it generated the plot. Anyways, so since I'm following the same tutorial, right, you will also get the same plot, so I'm not drawing it again. Instead I will explain the code. So next what I have drawn, tried here, like just to show you how the data changes from raw count to normalized count to scaling and top 100 genes. And since scaling will be applying on the gene level, right, so I'm just showing you the top 100 genes. Okay, and yeah, and we need to transpose it, so that because genes are there on a row, right, so we need to transpose it. So I'll show you. Already I have made a plot over here. I'll just show you. So each axis is one gene. So now this was the raw count, right? So you can see the count, the scale was 0 to 40. So after we normalization, the gene looked like like this. Okay, but after scaling, now you see the data is scaled and we have 0. So all these genes, the mean expression is 0. And okay, and you will have negative to positive. Okay, so that means in some cell that gene is less than that mean in expression, and in some cell that gene's expression is higher than the mean. So now you see that all I have brought such that the scaling makes that the centering around 0, mean is 0 of all the genes, and standard deviation is 1. Okay, and observations will be some x standard deviation away from that mean. But so now because of scaling, all the genes are comparable. Okay, so that's all I think I had here. And yes, so what we learned now, so in this video we talked about scaling, and we saw that scaling needs to be done on per gene level, and we are doing a very simple standard normalization, so centering around 0 and making standard deviation 1. And yeah, so before that we identified the top 2000 highly variable genes, and on that we will apply the scaling. Okay, so now what we have finished, so we first removed the noise from the data by QC, then we normalized cells vertically, then we normalized genes, or let's say scaled those genes horizontally. So now our data is perfect. Okay, so it's noise free and it's scaled and normalized. So now we can go ahead with the next step in the pipeline, which is dimension reduction. Okay, so once again, why we need dimension reduction? Because so many genes we have, right? So 13,000 genes we have, so we cannot go and analyze those many genes. Okay, it's very difficult. Okay, and lot many redundant information is there in data, which PCA will help us to reduce the dimension by, and also remove those redundancy. So we will see why we need to do PCA and what is PCA in the next video on like a dimension reduction, which is the next step of the Seurat pipeline. Okay, yeah, thanks for watching this video. I hope this video was useful. If yes, so again support my channel through like, subscription and press the bell icon.